Hello, everyone. Welcome to watch the video produced by Creative Biolabs, who has extensive experience in various antibody production and engineering fields. In this video we will study the overview of membrane protein and membrane protein antibody. Membrane proteins represent about a third of the proteins in living organisms. Based on their structure, there are main three types of membrane proteins. The first one is integral membrane protein that is permanently anchored or part of the membrane. The second type is peripheral membrane protein that is only temporarily attached to the lipid bilayer or to other integral proteins, and the third one is lipid-anchored proteins. Here we only describe the first two types of membrane protein. According to their relationship with the bilayer, integral membrane protein can be classified two primary types: integral polytopic proteins and integral monotopic proteins. Integral polytopic proteins are also known as transmembrane proteins, which can span across the membrane at least once. The first three types in the picture are common forms in integral membrane proteins, such as transmembrane alpha helix protein, transmembrane alpha helical protein and transmembrane beta sheet protein. Integral monotopic proteins are one type of integral membrane proteins that are attached to only one side of the membrane and do not span the whole way across. There are four types of interaction between integral monotopic membrane protein and cell membranes: by an amphipathic alpha helix spiral, by a hydrophobic loop, by a covalently bound membrane lipid and electrostatic or ionic interaction with membrane lipids. The latter four types are listed here. Membrane proteins play crucial roles in all organisms, where they serve as, such as, membrane receptors, ion channels, GPCR G-protein coupled receptors, and various kinds of transport proteins. As you can see, there are 5 kinds of functions. For example, the first is intercellular joinings. In an organism, different cells can be joined by membrane protein which can perform a series of biological function. The second function is enzymatic activity. The third is transport biological material by performed membrane transport proteins. The fourth function is that recognize cell to finish primary or complex biological mechanisms. The fifth is signal transduction, which plays a vital role in organisms. Compared with soluble cytoplasmic proteins, a variety of difficulties in expression of membrane proteins. 1. Membrane proteins are not just released into the cytosol but must rather be targeted and translocated to their final destinations in membranes. In particular, in eukaryotic cells, there is need a more complicated biological process. Requires sophisticated recognition and sorting mechanisms. 2. Copy number and capacity of prokaryotic and eukaryotic expression systems are limited because of translocation machineries, and translocation can be selective only for distinct groups of membrane proteins. 3. For structural and functional studies or other purposes, it is have to extract membrane protein from cellular after expression, followed by transfer into artificial and defined hydrophobic environments, like micelles or liposomes. Expression of membrane proteins in expression systems / cellular background, as closely related to their origin as possible, may be the most reasonable strategy. However, the efficiency and workload of the individual expression systems are quite different, and preparative amounts of membrane proteins are often only obtained with bacterial, yeast or cell-free systems. In this part, basic optimization parameters characteristic for the individual expression systems are illustrated, and the most critical parts are indicated. Some optimization parameters are not considered. Vector or target design. For example, fusion technologies or modifications of the expression cassettes. Gray bars illustrate the success in obtaining membrane protein structures after using the individual expression systems, while white bars correspond to those membrane protein structures obtained only after recombinant expression. Membrane proteins represent the vast majority of clinical drug targets, and are a focus of pharmaceutical and biotechnological interests. Despite significant and considerable recent improvements, the expression of functionally folded membrane proteins in sufficient amounts for functional and structural studies is still a challenging task. However, there are still many strategies for preparing membrane proteins that have been developed by specialists from Creative Biolabs. In terms of anti-membrane protein antibody discovery and production, Creative Biolabs is able to provide various methods, from immune antibody library construction by phage display to native antibody discovery by antigen-specific B lymphocytes, cytometry technology. If you are interested in learning more about how we can help with your projects, please don't hesitate to contact us at info@creative-biolabs.com or call us at 1-631-871-5806. We will be more than happy to serve you.